So I'm going to continue on the subject of using super-resolution imaging to look at the biological questions. In this case, I'm going to tell you the work we have been doing on imaging and characterizing the axonal cargo transport in neurons. I want to acknowledge that most of the lab work is done by a very talented PhD student in the lab, Yi, who's going to present a poster, and also she's assisted by two undergrad students, Stacy and Brianna. And some of the computation work, which you will see in a moment, is done by another PhD student in the lab. And we couldn't do this without the help of all our collaborators of the QSTORM project. And Jessica's lab provided us with the quantum dots. Peter's lab did all the storm imaging for us, and we exchanged expertise with Beth's lab. And finally, Carol played a very important role of coordinating the project and maintaining the web. Okay, so I want to start with telling you a little bit about the basic biology of axonal transport, and specifically what we're interested in is in its regulation mechanism by a microtubule-associated protein tau. And after that, I will tell you a little bit about the imaging work we have done, especially using storm imaging. And then I will tell you the biological findings we made so far, and I'll give you a discussion and outlook. So neurons are really polarized cells. What is very special about neurons is if you look at neurons, the structural hallmark is a cytoplasmic process of which is called axon. And in our body, this process can extend actually more than a meter at a diameter of a few microns. So the protein synthesis and degradation is mostly done in the cell body. So obviously, you can see that there's a logistic problem, because to maintain the metabolism over this very extended process, then the cells have to do something. And essentially, the cell takes the same strategy as we use in our society, is to develop a highway system. And what you see here is actually a very nice electron micrograph, another one, taken by Nobutaka Hirokawa about 30 years ago, showing that the transport of different organelles along the microtubules, and some of these we believe are mitochondria. Now, the reason we're interested in this process is that first of all, it's critical to the survival and function of the neurons, but also from a more general biology perspective, and because of the geometry of the neuron, this gives us a highly simplified system, as you will see in a moment, which makes our life much easier in imaging. But also, this provides a very, very powerful system to study the intracellular transport in the more general context, meaning that what we learn from this will tell us more about this process in non-polarized cells. So I want to start with an example that I want to show you how this works in Drosophila. This is the third instar larvae. And this is what you're looking at is the transport within one axon, the one small window along the axon. And this scale, this larvae is about 3 millimeters, 3,000 microns. So let me see if we can play this. So you can see some of the particles that go in towards the distal and to the synaptic terminals, others back, returning to the cell body. So you can see the bidirectional transport process. Now, what we know so far is that this process is driven by the molecular motors. So the transport towards the distal end is driven by different kinesin families. What I'm showing you here is the kinesin 1, conventional kinesin. The transport going back to the neuronal cell body is driven by the cytoplasmic dyning. And the function of cytoplasmic dyning in vivo requires a cofactor, it's a dinactin. So this is essentially the molecular motor machinery. That's not the focus of the subject today. Instead, what we're interested in this study is to look at how this process is regulated. Now, if you look at the configuration of all this molecular machine, and you can see that the regulation, at least there are different pathways you can think of. So first of all, it can be at this interface, meaning that the docking protein, the docking between the molecular motors and also the cargo. So meaning that you can control the unloading and loading and the unloading of the cargo. So that's one pathway. The second pathway is you can use for, different kinases can regulate the molecular motor functions. That presumably may regulate, modulate the velocity and direction of the transport and so on. And the last pathway, which is the pathway I'm going to focus on today, is the interaction between the motor domain and the microtubule. So this is essentially the strategy used in our railway system. So we're looking at this pathway and specifically looking at how the microtubule-associated protein is going to modulate this process. So in this case, now, the protein we select is the tau, is the microtubule-associated protein. It's most abundantly expressed in axons. Now, in human brain, it has 6 isoforms, so the structure of the protein is very interesting. So if you look at the C-terminus, it has different multiple domains of microtubule binding domain. So shown here, the different isoforms have different numbers. But also has the N-terminus is actually because of electrically negative charges project away from the microtubule. So the way this protein works, at least from the electron microscopy studies, that they align with, they bind to the microtubule along the direction of the protofilaments. So you can see that if this is, now this is a very important protein because it stabilizes the microtubule. Now you can see that this also created a problem for the transport. If you think about that, you have motors carry all these cargoes passing by, then this can potentially generate, pose a roadblock. So how does this work? So far then the model we have is in the community is that the phosphorylation of this protein is actually reduces binding affinity to the microtubule, so allow you to temporarily detach, and then this dephosphorylation allow you to reattach. So this is kind of a dynamic steady state, so allow these allow these cargoes to pass by when they detach, and then it's going to come back. Tau is going to come back to the microtubule and then stabilize the filaments. Okay. And in this case, then, following the theme our first speaker talked about, there's also spatial heterogeneity in this protein, is that when you actually go from the cell body towards the distal end, there's a gradual increase in the concentration of this protein. How this protein modulate the intracellular transport is unknown. So that's the question we want to study. So I want to tell you a little bit about the experimental strategy we're using here. Is that we do this in Drosophila. We take 2 fly lines. One is expressing a amyloid precursor protein tagged to YFP. So these type of vesicles is being very well characterized, driven most by kinesin 1. Whereas we also look at a different type of cargo, is a synaptotactin protein tagged with YFP, and this is driven by another kinesin family, kinesin 3. And then we essentially look at a tau overexpression background. We look at 2 mutants. One is the inserted in a human wild-type tau. The other one is the fly line inserted with the point mutant human tau. And I want to mention briefly that these 2 mutants actually originally generated to model the Alzheimer's disease, and overexpression has been shown to cause neurodegeneration. But that's not the focus of our study. So what I'd like to do next, I want to tell you a little bit about how do we do the imaging, and then how do we quantify this process so that we can make biological findings. Okay. So the way it is done here is that we take the third instar larvae, we dissect it, and you can see that, for example, here is with fluorescent label all the nerves. You can see that they actually project out. So it provides us a very long extension of all these axons, allow us to do the imaging. Now, because of in the Drosophila system, there's naturally these identical bands, provide us with the landmarks, allow us to define consistently different regions, A3, A4, A5, and A6, and this allows us to test. Under the assumption that the tau has a spatial heterogeneity, whether they have the similar motility or the transport behavior in all these different regions. So if you put all this together, so this is actually similar to what I showed you before. So you can see the transport process is a very busy process. Some of the cargoes going this way, the others going this way. Okay. All right, so now why do we need nano-resolution imaging? Well, for many, many reasons. First of all, then, these molecular motors are known to walk at steps of nanometer sizes. For example, kinesin is generally walked at a step size of 8 nanometer. Dyning is more flexible, can be 16 or 24. But also, which is shown here. Here, what I'm showing you is actually the step movement of a dyning. You can see that roughly about 16 nanometer. But also for these cargoes, actually, not only think about this as a microtubule filament. Not only they go along the filament, but actually they also move sideways. This is actually a known effect. So for us to really very precisely characterize the behavior of all these individual cargoes, we need to get to the nanometer resolution. So this is why to use a STORM imaging is a very attractive solution. Now, the other part of this project is to use adaptive optics. So you can see that clearly, I don't need to explain this to you, is that this is a very invasive process. When we do the imaging, we have to cut it out. Okay. So essentially, then the flies, you have to give them away after an hour or something after you do the imaging. Whereas if we can use adaptive optics, then will allow us to keep all these flies in a non-invasive way, and we can actually follow their development. And this is particularly important to look at the pathogenesis of all this process. Okay. As Beth mentioned, that this is a really ongoing project, so what we did, the strategy we took is that we didn't try to start the STORM imaging in the flies because that requires the injection of all these quantum dots and so on and so forth. So we started with the cell system. More specifically, we started with a very robust cell system with the PC12 cell, very easy to culture. And then this is what is shown here. What is shown here is the PC12 cells, and after they induced by nerve growth factor, they grow the neuron axon-like structure, and not exactly an axon. And then first thing the strategy we took is initially we decided, okay, the most quote-unquote direct way is to inject, right? You couldn't be better than that. You directly inject the quantum dot in and you get it. And it turns out that took much longer than we expected. So what I'm showing you here is that it's barely visible here. You can see if you stretch your imagination, you can see that there are actually 2 quantum dots here within this cell. And that's pretty much the signal we can get. But what makes things even worse is that these cells die. Okay, so apparently there's some apoptosis factors being triggered and so on and so forth. So we did some debugging, and then we worked with Jessica's lab, who provided us with a new batch of quantum dots that are coated with my cells, which is the one that actually Gan talked about. And what we know now, there are many different ways to bring in all these quantum dots into the cells. Now, because of the coating of the micelle, then it becomes very hydrophilic, make them easier to get into the cell. This is actually what I'm showing you here is one case that one result is actually going through a very simple incubation and going through the endocytic pathway, and these quantum dots get into the cell. I have to say that the contrast is bad because then there's a fair amount of background fluorescence, but that's because of the co-incubation. So let me play the movie here. So you can see this quantum dots dance around, going through a fairly random, Brownian type of motion, which is something we would expect, because in this case, then they're not targeted. They're not conjugated to any specific antibody. They're not really meant to be, recruited to any specific cellular structure. But what this tells us, teaches us a very important lesson here is that, so what he's shown here is that we repeat this process multiple times so we can very repetitively, reliably bring this into the PC12 cells. Now, what this shows us is that there are different factors we need to consider in bringing all these quantum dots into the cells. And for example, in this case, then the incubation, the endocytic pathway seems to be a nice path, and go by conjugating by encoding all these quantum dots with the micelles. What we're doing now is actually to focus on the real neuron system, the mouse hippocampal neurons, which is the system we decided to use for the study. So for the next part, I want to tell you a little bit about the biological data we get. But before I talk about that, I need to tell you that how do we quantify the transport phenotype? For that, we need to do some computation. We need to essentially write software to track all this cargo. So this is done by a standard technique. What is shown here essentially is that this is the image signal of a quantum dot. And then what you do is that fit that with the point spread function of a microscope. This allows us, this is actually a technique fairly old, it was done in the 1980s, that allows us to localize all these specific particles up to a resolution of a few nanometers. So what is shown here is actually, everyone in this room can see that although this is the same bright spot, you can see that because of the different profile, you can see that this position of this spot, this bright particle is actually close to the upper left, where this is close to the lower right. So intuitively, this is how the point spread function works to resolve this location of this particle beyond the resolution of a single pixel. So this technique is a general technique. It's not, we can use that, combine that with different imaging modalities, especially the STORM imaging modality. Okay. So before we move on, we need to make sure that the software is doing exactly what we want it. So what we did is the following, is that we mount different fluorescent beads or quantum dots onto the cover slip, and then we move that on a piezo stage. And for this piezo stage, it has the capability to very precisely control the movement up to a step size of half a nanometer. So what we did is that we drive the stage, use electric signal to drive the stage at a stepwise fashion. For example, in this case, we drive it to move at a 100 nanometer step. This is shown by the red signal. And then we ask the software, what do you think? Okay, the software is going to detect the position of all these particles, whether these could be fluorescent beads or quantum dots. And the software is going to return what he thinks is the position in green color. And so you can see that it follows pretty well. And it did break down when the step size becomes very small, up to the 60 nanometer. Then we can see that the limitation of this software tracking technique, and so that you can see that the red signal is the ground truth signal, whereas the green signal is what the software thinks. But by and large, then we make the conclusion that our software can follow the transport of up to a resolution about 10 to 20 nanometers. So with this technique, what we did is that we need to track all these vesicles along the axon. So the way it is visualized here is that this is what is called a Kymograph. So what he shows is that you take a small band of pixels along each frame, and then you put all this band of pixels together from frame 1, frame 2, and frame 3, and so on. So this is just a way to visualize the transport of different vesicles. For example, let's say this one, it actually its position over time is represented by this trace. So this axis is time, and this axis is position along the axon. So you can see this represents one vesicle going actually towards the distal end. For example, this represents another vesicle going back to the neuronal cell body. For those ones that are more or less vertical, those are ones that do not move much. Those are stationary ones. So when we actually look at all the flies we have and look at the APP vesicles, so you can see this is control. You can see, for example, there are a fairly large fraction of particles going towards the distal end, towards the synaptic terminal. Whereas when we overexpress the human tau, you can see that the motion, the transport is significantly impaired. And you see a lot of stationary vesicles. And in another genetic background, when we look at the point mutant tau, you can see that in addition to the increase in the stationary ones, there are actually very few going in this direction, and it seems that the transport is actually reversed and going in the backward direction, going back to the neuronal cell body. And this is we did this looking at different regions. You can see in region 4 and A5, and then you can appreciate that there's actually regionally there's some heterogeneity between different regions. So there's more data than the time allowed me to present here, but I want to present this. I want to show you that, for example, if we quantify the fraction of stationary cargos, this is actually readiness and control. You can see as you go gradually from the A3 region towards the distal region, there's indeed a spatial heterogeneity. As you go towards the distal region, the A6, you get more stationary ones. And this is actually repeated in the mutants. For example, in the point mutant, overexpression of human point mutant tau, and you can see that there's a further increase in the stationary cargo. So this is consistent with the existence of a gradient. And then for the others, it's roughly the same, but there are exceptions. But what we also learn from this is that actually this is not generally, the transport impairment is not generally true for all the cargos. So what he's shown here is that there's a significant decrease in terms of the transport velocity in the APP vesicles, which is characterized to be driven by kinesin 1. Whereas for the kinesin 3 cargo, the synaptotachin vesicles, there's very little, very mildly impairment. There's almost no statistical significant change in the velocity. By the way, this star represents a p-value is less than 0.1. Okay. So there's a very little change in the transport of the other type of cargo. So this tells us really this is the regulation of all this transport process is cargo specific. And then if you think about what I've told you so far, then this is also region dependent. Okay. Now, when you look at all these neurons within the Drosophila, you will very easily notice one thing is that, for example, this is the control. You can see that you see the buildup of all these different clocks. For example, the Y-type tau, you can see those buildup of all these vesicles. These are traffic jams. And then in the point mutant form, it's actually even more severe. You can see that there's a more buildup, more clogging. And this is what it is is a zoom in of one of the regions. Now, when you look at this, you will notice that actually there's a spatial kind of a separation between these clocks, and they kind of follow a more or less a uniform spacing. And so we wonder that what drives this, and then it doesn't really take us too much to think about that. This could be that due to the accumulation of the tau protein. So what we did is we did the immunostain, and we look at the tau. So for example, this is the mouse hippocampal neuron. So you can see that the tau does form all these small clusters along the region. And you can see also the signal is quite diffusive. It's not surprising you have all these taus in the cytoplasm, in the exoplasm. But this really doesn't really tell us much. Okay, so for that then, so we we collaborate with Peter's lab, also with the grad student Andrew, who did the work using the STORM imaging. In this case, then, is Alexa 488 with labeled tau. So this is actually a STORM imaging of a region within. This is in this specific case. This is from a PC12 cell. So what you can see from this is actually telling us something very exciting. Is that first of all, then it resolved the signal at a much higher resolution, but also tell us something very interesting. For example, these different classes of taus may likely sit on different microtubules, but they also these different tau clusters, and presumably they actually may play a role in actually informing different bundles along these microtubules. So these are very important information, tell us how the distribution of the tau may actually mediate the organization of the microtubule and so on, which this information is you cannot get from the confocal image data. So I want to wrap up and tell you, just to summarize that what we have done so far is that we have done a pretty comprehensive characterization of microinjection, and unfortunately the outcome is negative so far, but that doesn't mean that this is a technique that's going to fail. It's just that we haven't find out the best way to do this, but we have a very comprehensively characterized it. We have found ways to successfully deliver the quantum dot into PC12 cells. We're doing more work on mouse hippocampal neurons, and then and also the Peter's lab, in collaboration with Peter's lab, we have been successful in using STORM imaging to characterize the spatial distribution of the tau. Now, from the biology side, so we have done a very comprehensive study of axonal transport. Unfortunately, I only have a little bit of time to talk about the results. But what we learned is that tau modulated cargo transport, preferentially it affects actually the kinesin-mediated cargo transport, meaning the transport going towards the synaptic terminal. And this modulation is consistent with the spatial gradient of tau. It's a region-specific, but also is a cargo-specific. Okay, so where do we go from here? What we're going to do next is that we're going to further establish, in collaboration with Beth's lab, and also establish a method to really deliver all these quantum dots into different cells. And also to establish and test all the switchable quantum dots for STORM imaging in fixed cells. Here, I want to comment that originally our strategy was to go for live imaging, and we realized that actually it's very helpful to do some fix and stay, because this essentially allows us to test absolutely almost everything we want to know. For example, you can test the switching of the quantum dots, to test the STORM imaging, to test the adaptive optics. So we believe that this actually Beth's lab came out with this strategy, and this is a very, very powerful change to the design, and which allow us to test all the techniques we have with very relatively low risk. And finally, of course, then the goal is that we're going to test the switchable quantum dot for live imaging in both the neurons and both flies. Now, in terms of biology, what we need to do, as you can probably already see here, is that we need to quantify the tau distribution, which is revealed by the STORM imaging, to really to really quantify this process, to have a rigorous understanding of this distribution of this protein. But also, we want to use this technique, again, follow the same paradigm using Beth's lab to look at the organization, spatial distribution of many other regulatory proteins to see how they're spatially distributed, many different kinases, different phosphatases, and that may potentially play a regulatory role. And finally, then we want to be able to analyze the behavior in live imaging data. I want to point out that because of the time, we didn't get to talk too much about the biology data, but we have a poster. Yi is going to present the poster here. She's going to be able to tell you more about the biological findings we have. Okay, with that, that concludes my talk, and thank you for your attention.